the transcription factors is the ultimate goal of these, what we call signal transduction pathways. Many times these transcription factors are either available in the cell, but cannot go into the nucleus, okay? So the cell will make these transcription factors in the cytoplasm, but keep them out of the nucleus. And the only way that these transcription factors can come into the nucleus to start transcribing genes, when they're induced, when the cells are induced, is when they receive a signal from another cell, which is the induction process, essentially. So a lot of times, remember I told you how sometimes cells will make proteins, and then they'll destroy those proteins. Well, this is an example. We're going to show you a couple of examples of how this is done, where they'll make these transcription factors, but they'll eat, and then they'll destroy them. But they're in the process of constantly making these until the moment that the cell gets some signal from another cell. The moment it gets that cell, boom, the protein stops being degraded, it comes into the nucleus, and immediately the cell can respond to that signal. That's one of the reasons why the cell would create these transcription factors, only to destroy them so that it can be available in a moment's notice, because there are certain things that have to happen almost instantaneously in development to be able to respond. It can't wait half an hour or an hour to start making these and then say, okay, now I'm ready to respond to this signal. So many of these pathways that I'm going to talk about are proteins that are constantly being made and set up so that at a moment's notice, a cell can respond to an inductive signal. And this is where differential gene expression comes into play. When it gets a signal, then these transcription factors are allowed into the nucleus, and then certain genes are transcribed based upon the inductive signal. Okay? Signal transduction pathways. That's what we're going to spend the rest of the time on. We may or may not get through all of this. This is where a lot of your questions are going to come into play as well. Because from here on out, when we start talking about vertebrate development, you're going to see these genes all the time. Sonic hedgehog, Wnt signaling, Hux genes, all of these types of things that come into play. So in almost all these cases, you essentially have some protein, which we call a ligand. So in the case of a protein that is secreted by another cell, that morphogen gradient, there's a specific name for it. We call it a ligand. Okay? Ligand is a term you're going to have to know. So the ligand, or the protein, and then the receptor that's found on the surface of the cell. The receptor, again, this is where competence comes into play, must match the ligand. So the ligand and the receptor have to be compatible. This is where induction and competence ultimately come together. The ligand is the inducer, or the protein being secreted by the inducer, and the receptor is the cell's competence, or ability to respond to that ligand. Ultimately, it's not about the ligand receptor. It's about what happens once they come together. It's about the internal cellular processes of the responding tissue and what gene transcription, or sometimes repression, comes into play. It's not always about turning genes off. Sometimes it's about turning genes on. It's about turning genes off. Okay? Because as we talked about during the commitment process, where they become determined, it's not just about some genes being turned on. Some transcription factors will turn some genes on and shut other genes off at the same time. So there's dual purposes in some of these transcription factors. That repression ultimately makes it so that the cells stay the way that they are, that they're going to become skin even if they have other signals coming into play. So that repression can sometimes shut off a receptor so that the cell is no longer competent to the signal. You see how complex this can start to get. Okay? All right. Now, again, I mentioned this before, is that there's only so many proteins that are made in nature. And what's fascinating is we know a lot about how humans develop, not because of us studying human embryology, but because we study all these other organisms which have the same induction methods that we do. They use the same proteins that we do to make the heart, to make the kidney, to make our teeth. So even though we have different genetics, the proteins that cause hearts to form in all vertebrates is pretty much the same, or proteins that are used are almost the same. Now, yes, there are differences between, say, insects and vertebrates because of the dynamics, but in fact, we do have similarities even there. Even though the proteins aren't exactly the same, they are very closely related. We'll talk more about that in evolution as far as paralogs and orthologs in terms of the proteins in evolutionary development. So the same proteins. As we study vertebrates, fish and birds and other mammals and things like that, we are actually studying ourselves because we use some, pretty much the same proteins in a lot of these processes. These are a lot of the dynamics that go into signal transduction pathways. You actually have these huge families of ligands and receptors. Like I said, of the PAX genes, there are 8, 7, 8, I think at least 8 of those. Hox genes, there's 4 main groups, but then there's subgroups within those. You've got Wnt-frizzled family, where you've got, I think, 14 different combinations and whatnot. So there's a great amount of complexity as far as ligand receptors. When you look at the receptors themselves, you actually don't just have one receptor. Sometimes you got to have the right combination of 2 different receptors, and this is where we have co-receptors and dimerization come into play. Sometimes the receptors are the same, which is where the homo comes in. Hetero is where the receptors are different, yet they must both be present for the cell to be able to respond to that type of interaction. You don't just, in some of these pathways we're going to go through, sometimes there are like 3 or 4 different ways that the same signal can go through. So, we have what we call the canonical Wnt pathway, and then we have the non-canonical Wnt pathway. And so we've talked about sequential induction. Again, the history of the cell. In one period of time, a certain signal transduction pathway tells the cell to do one thing, and then a little bit later, because of this sequential induction, the same pathway can tell it to do something completely different. Okay? So again, it's not as simple as saying, oh, this one does this all the time. Nope. In this circumstance, at this particular time in the cell's history, it might tell it to do this. At another time in the cell's history, it might tell us to do something completely different. An example of this is when neurons are finding their muscle or their target where they're supposed to go. In some cases, the neuron has to, to go down its pathway, it receives a certain signal, and then at a certain point, all of a sudden, that signal doesn't repel it anymore, it actually tracks it, it comes across, and then it goes through a certain point, and then it repels it all over again. So it matters as far as when, where, and its overall history on how the tissue is going to respond to that same signal transduction pathway. Competitive and inhibitory regulators. What this means is there's a lot of morphogens, gradients and such, that can actually compete with one another. When you have 2 different gradients hitting each other at the same time, sometimes the proteins can interact and completely cancel each other out. So there's competition between some of these signals. And that's a good thing because it creates different levels of a morphogen that the cells can actually respond to. It's not just high to low levels. Sometimes you get this crosstalk between them, and you have different types of inhibition that regulate how much morphogen they're being is attaching to them. And then, of course, finally, sometimes the cells are their own inducers, where they start something and then they just kind of keep the cycle going. And it requires another cell to come in here and say, okay, stop. There's this positive feedback loop sometimes with some of these signal transduction pathways where they'll just keep, once they've been triggered, they'll just keep going without any other stimulus. And then they need something else to come in and say, okay, stop. You don't need to do that anymore. There are 4 ways that cells can communicate with one another. The main one that I've been telling you about is called paracrine signaling. And this is the one that we're going to spend most of our time on in development. What is paracrine? It is when the proteins from the inducing tissue are secreted out into the extracellular space, where you have this diffusion coming out from the inducer cells that then spread out and start affecting all these other tissues. That's paracrine signaling. So paracrine is when the protein is secreted from these cells. Juxtacrine is where the inducing ligand is actually bound to the membrane of the cell. This restricts its ability to only induce adjacent tissue. So it doesn't actually secrete the protein. It makes the protein, and it is usually a membrane protein. So juxtacrine is where you get just cell-cell contact, and the prime example that we're going to go through here is notch signaling. Most everything we're going to go through today is paracrine signaling, but notch delta signaling is an example of juxtacrine, which is used in nervous system development. Autocrine, this is when the cell secretes a signal and it induces itself. So autocrine can be paracrine in nature, but instead of inducing other tissues, it just induces itself. So we're not going to talk too much about autocrine. And so the difference between endocrine and paracrine is that endocrine is when the organism, typically those that have a cardiovascular system, secrete them into the cardiovascular system, travels through, and it induces other tissues through that mechanism. So that's the main difference here, is endocrine is through the blood. Paracrine, you can see here in this example, it's secreting proteins. This creates a gradient, a morphogen gradient. Juxtacrine, they can either have the extracellular matrix, which is a series of proteins that can induce the tissue, or you can actually have physical contact between one membrane and another membrane of those. Those are both juxtacrine. And then autocrine and endocrine. We went through those. So let's focus on paracrine and juxtacrine. Those are the two that I'm going to want you to explain to me and be able to describe. This picture just kind of shows the same thing of the different types of, how you have autocrine and it just keeps signaling to itself. Or if you have, here's an example of a reciprocal induction of paracrine signaling, where one secretes a protein that induces this one secretes a protein and induces that one, and they just keep going back and forth. You'll see a good example of this too later on. All right, paracrine signaling.